Well, thank you very much for this invitation. It's really an honor. And it's also a pleasure to present this project, B4EST, which many of you probably know and even probably have worked in. I really apologize in advance for the simplifications I had to make to explain so much in so little time. I'm not asking for extra time. I know that you have a very tight schedule. So let's go on. B4EST, so the project B4EST started in 2018 and ended officially a year ago, approximately September last year. And it was coordinated by Catherine Bastian, whom many of you probably know. The title of this project was Increase Forest Survival, Health, Resilience, and Productivity under Climate Change and Natural Disturbances, while maintaining genetic diversity and key ecological functions. So behind that title, I think there is a whole landscape of researchers that were working in 90 different institutions, partners, working with 8 different species. And behind all these species lies really a highly heterogeneous reality, as you can imagine, with very different breeding programs, socioeconomical context, also biological constraints and knowledge. And also, something that we have to say it's unfavorable when it comes to forestry is really this ratio of researchers to species. So, and it's because of this complexity in such conditions that it's really important to do collaborative work like the one we did in this project. Before us, there were other collaborative projects working also on forestry, and also we know that there is also other projects that came after before us that are continuing with this collaborative effort. So, just to give a brief concept about the project. Well, of course, the center, as you probably know, genetic diversity. That's really the main object, the main object of study, and everything gravitates around that aspect. And here we have 3 different angles of study that are really characteristic of this project. One, which is the resilience to climatic change. That was one of the the main angles of study of the project. Another one was the trade-offs. So all the constraints that affect the co-evolution or the co-selection of traits. And so that have to be taken into account whenever we do some kind of management. And then we have also this risk and benefit analysis, which is really important when we deal with the management of genetic diversity in selection conditions. So there are these 2 actions, forest management and tree breeding, that are connecting these different angles of the study. We mentioned diversity, and we see already this diversity in terms of the species, and in terms of the coverage of the forestry landscape in Europe, in terms of socioeconomical aspects, but also in terms of we have species that were threatened, we have species that are very important economically, etc. And this complexity was really studied by really a big group of researchers that, as we said, were represented by these 90 different institutions, and really with a multidisciplinary research panel of expertise, of skills, not only genetics, but also, ecophysiology, pathology, entomology, etc. So that was really one of the richness of the project. So how to deal with such a complexity? What was the strategy? Well, a simple way to show this is, of course, try to split things in order to attack from different angles of view. And so we have different work packages. You see at the bottom, work package 1 and 2, that are really those that are producing scientific knowledge. Work package 1 more on key abiotic and biotic factors that affect the tree responses, or the identification of genetic diversity that is relevant for these tree responses. We have another level of work packages that we are dealing with, using the information coming from work package 1 and 2, but using technology and methodology to integrate that information in order to study the deployment or to study the tree breeding. And again, there were another level of work packages that are even more integrative, that were dealing, for instance, with communication, but also with the impacts of innovation that came from the project. There is another way to present things that probably is simpler, that will help us to present the different kind of works we have done during this project. And it's represented here by this kind of pie chart with three main sectors that represent the different phenomena we are studying. Behind phenomics, we have all the tree responses that we can measure, that we can study, the phenotypes. We have embryonics, which is all the different environment characterizations that we do in order to know better what the conditions, the environmental conditions, biotic, abiotic, where the tree grows, and also genomics. Behind genomics, we could have many other, let's say, endophenotypes that lay between the genome and the phenome, and that are related to the genomic architecture of the trait. So all this is studied at different levels by different work packages, and I will try to show that very, very rapidly. So in work package 1, mostly we were dealing with the interaction between phenomics and embryonics. In work package 2, it's the links, the associations between genomics and phenomics, or between genomics and embryonics. In work package 3, we were integrating different pieces of information like phenomics, embryonics, and genomics in order to improve the prediction of the trait, and also to give extra selection precision to the program. And then we have the other two work packages doing more scientific level of output that we're dealing with mostly with prediction and projections. Work package 4 in terms of the dissemination of the genetic gain, and work package 5 with the risk benefits analysis. This is it's not possible to talk about all these, so I will concentrate basically on the first two work package, on the first three work packages, which I think present more the kind of scientific papers that could interest the public we have today here. But we have done very interesting things also in work package 5 and 4. For instance, dealing with the planter's guide, which is a guide for foresters in order to choose the good material for a good spot in the forests. We have also simulators like Blueberry 2. That it's a very good tool for silviculture, or with also some economical analysis on the impact of innovations in tree breeding. But we are not talking about those today. So we will start with the first series of work that deal mostly with the first set of information that we require in order to study tree reaction. So, which is related with embryonics and the link with phenomics. So, the first, let's say, pillar that is required here is really climatic data. So, we need really to characterize the tree environment in order to make predictions. And so, climatic data is really a key source but often this data is scattered over multiple databases. So one big effort within the project was really to provide with this raw data with an easy access, together with a series of tools to query or to extract or to represent this information. And this is Climate EU, which is really, well, a database that is able to provide this kind of services. Well, the main service, the main feature of this Climate EU is really being able to do a downscaling to generate scale-free data. And for any kind of location, accounting for orography, for changes in elevation, for very many climatic variables that could be produced annually or monthly, with historical records for more than a century, but also for future projections. So this is extremely rich tool for any kind of a study, and it's been used in the framework of the project, but also for other people. We have here an example with the mean annual temperature, with the kind of map, the kind of output we can get, but also, of course, with the raw data behind. Another tool, which is also quite interesting, is this climate matching tool, which actually locates places that have a climate that is close or similar to the climate of a given place, even looking forward or backwards in time. And so it has here to the left, we have, well, the main entrance of the program, where we have, well, a very, very easy way to do the settings. And we have an output that we present here with a colored map. And it's basically an example where we are showing the location of places that have a climate currently that is similar to the climate that Brasov will have in 10 years' time. So we have different colors, and actually the darker color is closer to the match. Okay, so this is also very useful, for instance, when we try to look places for trials, or places that are similar to our trial, for instance. Well, one of the first examples of of the use of this climatic data we have here in this nice paper, where the authors try to address one long-standing open question. With, so far, not a lot of empirical evidence. And this question is really, the factors that influence within population cognitive genetic variation. And the authors assume different hypotheses. Classically, there could be the admixture between several populations, or the fact of having, a harsher environment, or the fact of having a very heterogeneous distribution of environments. So all these could have a role in the genetic diversity we will have in the population. And so they were using characterizing the environments for a series of common gardens that are represented in this map. Common gardens, the populations that were contained in these common gardens, and we see with these colors, it's the modeling of the climate in terms of summer droughts and winter cold extremes that we have for the different populations contained in the common garden experiments. So the kind of results, very briefly, very simply, is that, actually there was no association between spatial and temporal heterogeneity and the fact of having, different genetic variability for the different populations. Also, there was no association or clear association between the admixture and the fact of having, different genetic diversity within the population. But there wasn't some interesting result in the sense that there was empirical support for that the genetic variation for early height growth was lower in populations that were experiencing colder winters. So these results really provide an empirical evidence of the potential role of natural selection in reducing the genetic variation. So, this is a kind of assumption, general assumption that everybody uses sometimes, but it's the kind of evidences are not so frequent. So another experiment using this climatic data comes from this review, this paper that really reviewed the uncertainty by given certain climatic projections by different models for extreme populations in Scotland. And so in this study, they need to identify the conditions that were the drivers of the population responses that were based on tree height and by using common gardens that are represented. The scope of these common gardens are represented in this map that we have collected. So the authors did a series of mixed models in order to retrospectively assess how height for the different populations has responded to the environmental variation. And this calibration by these models was used to assess height against climatic projections. And by looking at the uncertainty among these projections, for instance, using the coefficient of variation, they were giving a kind of inference about this uncertainty. And so what I did find is that while the drivers were different in the two extremes, that's something, it seems logical, but it has to be found. So in Spain, height is mostly driven by drought. And when we look at the climatic projections, we see some stagnation or loss of height over these future projections, but with very low uncertainty. So there is very low variation between these projections. And when we look at Nordic countries, those height mostly driven by photoperiod. And when we look at the climatic projections, there was an increase in height, but there was a high uncertainty associated with that increase. So authors pinpoint the fact that maybe some of the uncertainty comes from extrapolation that was needed, and so there is a need for denser data. So the key messages, the importance really of considering the environmental characterization in the assessment of the vulnerability, and the paradigm shifted from the last century. So where environment was a kind of nuisance that we have to remove to correct by using average performances, to what we have nowadays, the efforts to integrate the fine characterization of the environment to bring more explanatory hypotheses to treat adaptation and evolution. And that's really something important. So another let's say bunch of outputs from the project came from the genomic part, and they are related to the fact of producing nice genotyping tools, genotyping arrays. And the strategy with trees is that some of them, especially the conifers, they have complex genomes. And in order to study those, there are usually multiple initiatives, and so it could be interesting to benefit from all those different, let's say, initiatives, different projects, but also to be able to discriminate all this information and keep the one that is really meaningful, really relevant. And so the strategy that we used here, it was a two-step strategy, in order to end up with a final array with relevant markers. With a first step, we used really a high density screening array, where we had the room, let's say, to put a lot of markers, and we could test those all these markers against a large, almost 500 diversity panel, in order to see the behavior of all those markers and keep the ones that are really of good quality. And that was the work that was done with a lot of filtering in order to get into the second step, in order to, to get really this 50K SNP with very high quality markers by doing, comparison with previous genotyping tools, by doing, of course, Mendelian tests, by doing a good distribution across the genome, etc., and many other, let's say, filters that gave at the end this 50K SNP. That was the one that is used for the different species. And within the project, there was also the finance to genotype with this final array more than 8,000 samples per array. 3 of these arrays were produced in the project. So the first one, it was the one that was produced for Scott spine. So here we have actually a comparison of the different, let's say, SNP qualities for the screen array and for the final array. And you see blue is the most quality SNPs, and red are those that are, to be rejected. And so you see that there is a big change with this 2 steps procedure in order to improve the quality of the SNPs that are included in the tool. So this is one species 50K array produced by this technology through a really collaborative work with multiple sources of genomic data, some capture, previous arrays, sequencing, etc. That was validated validated in terms of performance using diversified population samples with a good discrimination between Finnish and Scottish populations, and also with low rates with parentage estimates. We see here this matrix with families in order to validate the parentage against the pedigree expectations, and also with low level of Mendelian errors. The second array is also an array for a single species, Norway spruce, but with a particularity that at least 50% of the probes were working with older spruces, and so that's a good feature. It was the first time a whole genome-based SNP array in conifers was produced. With a panel of 29 trees that were collected in Northern Europe, and again with the characteristic of having low error rates, parentage estimates that are consistent with expectations, and low level of Mendelian errors. And the final array, it's a little bit different in the sense that it involves 4 species. And so it's 2 pines, pinaster and stone pine, poplars and ash. So it uses exactly the same technology with these 2 steps, and it has the particularity that it really brings a cost-effective genotyping tool to a very large community of users for a species with little chance, some of them, of developing a single species initiative. So it's really something that brings a lot in a very cost-effective way. It was really built through a very large collaborative effort across countries and teams, and also during the pandemics, and that was really tough. So it was developed from a very wide range of genomic resources. Here you have a table, but we are not looking into the details, with really all kinds of genomic resources for the different species. And the performance was validated using, very diversified samples, populations, etc. And again, with low error rates, good expectations in terms of parentage, and low level of Mendelian errors, talking globally. So obtaining these genotyping tools is one of the raison d'etre of these collaborative projects, really. These tools have been developed in a collaborative way, which is essential for some of the species with few initial resources. And also this same collaborative process has significantly reduced the cost of access and use, and that's really very important. So here we enter in another, let's say, space of the project, where we try to link, some of these genomic resources developed, to link those resources, this information with phenomics or with the embryonics. And we have here two good examples. In the first example, it's an example where really we try to work with the concept of genetic architecture, which is really important in the sense that it could give some insights on the potential population responses. There are two hypotheses that were considered. Either we have this polygenic adaptation model, which is basically something that is close to a kind of infinitesimal model with plenty of genes with basically single effect, and so that the change in population mean comes from a shift in the frequency spectrum. And we have another hypothesis that comes from the change in population mean happens because there is these selective shifts that are represented in this figure below, where you have these big jumps. I don't know if you see my mouse. These big jumps in allele frequency because of this rapid fixation of these mutations with a big effect. Versus this another, let's say, polygenic model where you have very many alleles that change slightly their frequency, and this makes the population moves in terms of mean, in terms of adaptation. So polygenicity is one way of assess to what extent or trait has a genetic architecture that is close, let's just say, to this infinitesimal model. So it's basically a percentage of SNPs with a non-zero effect for the trait. And this can be obtained by using genomic modeling. And this feature could be maintained in the population by negative selection that removes let's say the big effects, the big negative effects. And so this could be looked at by, for instance, estimating the correlation between the allelic effects and the minimum allele frequency. So in this study, they were using a common garden that is represented here in the map, and they were obtaining different estimates of polygenicity. And what we can observe is that these patterns among traits and among environments were quite similar. But the main important result is that there was some negative selection. So this correlation between allelic effects and the allele frequency for key traits, for height, for phenology, for SLR. And this correlation was sensible to the changing environments, and that's something that it's interesting. And actually, it pinpoints to the fact that current prediction models that are mostly based on these selective shifts could underestimate somehow this adaptative capacity. If in fact behind we have some polygenic structure that could act rapidly. Another paper. In this paper, the authors have investigated the role of different factors in the prediction of height in a network of common gardens that is represented here in the map. And for that, the authors, they were using a series of increasing complexity models by including different factors, explanatory factors. Of course, those that were already in the, let's say, design of the common gardens, like the corner repetitions, that gives some kind of a view of the plastic component. but also the differences within provenance and within provenance that gives some idea about the genetic component. But they were using some climatic regressors, but also some genomic information in some of the most complex models. This genomic information was used to discriminate gene pools, so to assign some of the populations to given gene pools, but also to add some extra information that came from a guez-based S&P contribution to growth in the form of positive effect allele counts. So it was just counting the alleles with a positive effect for the growth. And so, it's the main results in a very brief message is that, well, there is a big importance of the plastic component, the big ton of replicates, followed by this genetic component, but also that the best fitting models comprise, not only climatic regressors, so this climatic information, but also the genomic information, like this gene pool assignment, and also this positive effect allele. That were able to predict provenances for new provenances for a high polygenetic trait, like height. And this is really an open door to the genomic integration, to genomic and the climatic integration to really make larger scale predictive models. So the key message is that, this kind of work, but also other papers that were related to these work packages, is that, this inclusion of the genomic information, as well as all the components that inform on the genome by environmental interactions, is really a key element in modeling the behavior of trees at a larger scale. And also this type of modeling also allows to make a better link with the predictive approaches that are used at the scale of population or group of populations in the context of genetic improvement, because basically it's the same mechanics, it's the same kind of modeling, it's the same kind of information between populations where the same integration is used also to improve the candidate selection. So here we have another panel of research in the work package 3, in the work package where we were doing some kind of integration in order to improve the prediction of the phenotype by integrating phenomics, genomics, and also environments. We have two examples of that. Well, in the first example, it's an example where we try to use different what are known as endophenotypes. So omic information that lays in between the genome and the phenome. In this case, it was just two of those. It was the SNPs, the sequence, and also the transcriptome. And this kind of information could really inform us, give us some idea of all the pathways of interactions that happens between the genome and the phenome. And so it can give us some idea about the non-additive effects, the interactive effects like epistasis that happen between these two extreme layers of information. So we were doing a kind of a very simple integration of information, this genomic and transcriptomic, by using concatenation, which is represented there. We just use, put all the information in the same matrix with some kind of normalization. And then, we were studying really the advantages of this integration in terms of the change in effects in the loci during the integration. So when we compare, a single omic model with a multi-omic model, what were the changes in terms of allele effects? And also in terms of the functional typology of the affected loci. And some interesting results came out of this. So first thing is that, the integration advantage in prediction over single omics was primarily for growth traits, or traits that were closely related to the tissues that were sampled for the Irania sec. It was cambium. And also one thing that was probably the most interesting part of the results is that the disadvantage in integration came because of a massive reduction in the effect estimates of loci that were actually linked to EQTLs, to nodes that were highly interactive. And so this massive reduction gave room to other, let's say, minor effects that were given some extra explanation to the phenotype. And so it's important to take that into account, because it gives us an idea that, well, the redundancy is one of the main factors we have to control when we do this kind of integration. I don't know, in terms of timing, it's okay. So this is the last work I would like to show you, and it's about another kind of integration. In this time, it's using genomics, but also using some kind of environmental index in order to construct norms of reaction. So usually when we do evaluation, we use very integrative traits like growth, for instance. But the problem is that it's such an integrative trait that it offers really very few analytical possibilities to identify the causal environmental drivers. And so if we choose kind of longitudinal traits, like the ones we can get from incoming cores or from proxies of this incoming cores, we have a kind of historical growth. And so we can correlate what happens historically with, the evolution of the climate at the same time. And so genomics here, it brings some extra predictability when we need to interpolate or extrapolate unseen performances along this environmental gradient. So the thing we did here, you have the microdensity profiles coming out of this increment course. This is the longitudinal trait, the phenotypic trait in terms of a vector. And at the same time, we are developing different climatic indexes that were describing this, trying to describe this evolution. And with this, we try to describe a function, which is a norm of reaction, and to fit that function with a random variation approach with the general polynomials. So this is the result. So here to the left, you have, these norms of reaction. This is just the corresponding part for the additive component. So that's why it's a deviation from zero. So there is also, let's say, an average performance of the population, which is also plastic. Okay, so the environmental gradient derived, we used two environmental gradients, one which was a little bit more complex and was derived from an ecophysiological model, gave slightly better predictions. Although, the paper was not, let's say, instructed to optimize this let's say, environmental gradient. Though the random regression modeling using genomics outperformed the pedigree, and this is something we can see in this upper figure here with the blue versus yellow boxes. And with two different climatic indexes, and there is some advantage of the genomic counterpart for the best of the climatic indexes that we were using. But the most interesting part is that actually we could use different ways of allocating the phenotypic effort for the calibration, and that was really a key factor. If the phenotypic effort was more evenly distributed across the calibration, so predictability could be improved in a very significant way, and this is something we can see in this figure to the right, when we compare one, let's say, way of doing calibration with 50 individuals with full records and 50% individuals with no records, against another more, let's say, evenly distributed effort that gave, much better results. And this is something that mimics what we could do with research data, for instance, which is a very simple way of producing a lot of data with a minimum effort. So the predictive models used in genetic improvement can and should be improved by adding new layers of information that, in addition to predictive quality, favor causal explanation. As in the case of multiomics, if intelligent variable selection approaches are developed in parallel, that's really very important. So we need the mathematicians there. And in the case of norms of reaction trajectory predictions, with a new role for the environment in the model, if it can be properly characterized. So there is also a lot of work in there in order to characterize and check which index works good with the model. So some final and broader thoughts on B4EST. Really the importance of training young scientists. This was really a very key point, especially in high demand areas like cognitive genetics, where there is really a big lack of expertise in genomics and bioinformatics. For instance, with 4 PhD students only in the work package 3 and several postdocs. Going collaborative is definitely a strategy for developing costly applications like arrays, like genomic selection, etc. Genomic selection opens the door to data integration from heterogeneous sources, as it provides really the glue to efficiently link individuals, data points, etc. Collaborative bidding between countries. This is probably the best and only option to face future bidding challenges. Really, I know it's difficult. There is a lot of administrative and political constraints for that, but it's really the way to go. 2 points more. System biological approach are key to understand the responses of trees to an environment, often providing more precise predictions of phenotype and mechanistic explanations at the same time. This is the case of multiomics. There is still a lack of expertise on how to integrate data in a very efficient way. Systemic approaches to tree evaluation that include detailed characterization of the environment in which the tree grows, in particular using longitudinal data and high-dimensional phenotyping to assess trajectories of plasticities, are essential to accurately predict responses to future environments. And there is also a lack of expertise to develop and apply these systemic assessments under selection conditions. So thank you very much. Here you have a picture of, the last international conference of the project in Lisbon a year ago, and it was really a very pleasant moment. Thank you very much.
Thank you very much, Leopoldo. Very impressive work and quite a lot to show us all. Do we have any questions? Okay, I have maybe a very, if I meet a breeder, he will at once ask, what does it cost, for example, to use these technologies, and are they about to be used in any selection schemes, these SNP arrays?
Well, I think because of the consortium and because of the opportunity we had, 5 years ago, to join a North American consortium that was in touch with Thermo Fisher at the time. And so we could really have access to this technology with a lot of, demands at the same time. And so we really had a cost of development that was really very good. Nowadays, the arrays are commercially available. And I think that for one of these, any of these ways, we could get approximately $40 per sample. So that's a kind of maybe it's a little bit roughly, but between $36 and $40 per sample.
Any other questions? It seems like it's quite late on Friday afternoon. So I think we will say thank you also because time is running. Thank you, Leopoldo.
Thank you.